Hello students. Today we teach about the reproduction in flowering plant. You know that reproduction is a process in which plant produce their young ones or any organism produce their young ones. But here we study about the reproduction in flowering plants. First thing we study about what is a complete flower. A complete flower means if any flower contain four whorls or florals, it means calyx, corolla, endoecium and gynoecium, in which calyx and corolla are non-essential parts and endoecium, gynoecium are essential parts. If four whorls are present, the flower is said to be a complete flower or bisexual or dioecious flower. If any one essential part is absent in a flower, just endoecium or gynoecium, if one is absent, that flower is unisexual flower or monoecious flower or incomplete flower. If a flower contains only endoecium or stamen, it means it is a staminate flower. If it contains gynoecium, that is called pistillate flower. So here we start how these plants are fertilized, how these flowers can be fertilized and produce their young ones. So the whole event is divided into three parts: pre-fertilization event, fertilization, and post-fertilization. But in whether it is in plant or any organism. But we study first the pre-fertilization event. Pre-fertilization events only the gametogenesis and the pollen transfer can be occurred. Gametogenesis means formation of gametes through reproductive organs. In plant, the reproductive organs are stamens and carpel. Stamens is made up of anther, connective, and filament. Anther, which is a bilobed structure, contain two theca chamber, which is connected through filament by a connective. The filament is a long stalk which holds the anther. The main part of a stamen is anther because anther contain pollen grains inside its chamber. If we cut the transfer section of an anther, we find the structure of microsporangium. Microsporangium is made up of four layers: epidermis, endothecium, middle layer, and tapetum. The three outer layer, epidermis, endothecium, and middle layer, are responsible for dehiscence of anther. It means matureness of anther because when it becomes mature, it becomes rust and release the pollen grain. Whether the tapetum provide the nourishment to the developing pollen grains inside the microsporangia. The pollen grains are mature inside the sporogenous tissue. So how this process can be occur? It means how the pollen grains are formed. This process is called microsporogenesis. It means formation of microspore from microspore mother cell or pollen mother cell. This is called microsporogenesis. In this process, the pollen mother cells first divide by through meiosis and produce haploid microspores inside the sporogenous cells. This is whole process called microsporogenesis. Now we know how these microspores are very small, and this is also called pollen grains. The pollen grains have two layers. Outer layer is called exine and the inner layer is called intine. Exine is made up of sporopollen, which is extremely resistant and shine in the high temperature and also in alkalic condition. But these exine is absent or thinnest in some places. This part is called germ pore. It means that produce pollen tube. Axine is after the axine, the inner layer is called intine, that is made up of pectocellulose, pectin and cellulose. Inside the pollen grains that contain a large nucleus and with dense cytoplasm. The development of the pollen grains is taken two parts. One is inside the anther and the other is when it transfer to the stem. Stem carpel of stem. Thank you. All right.
Yeah, I like it.
आ गया